Histology is the study of tissues. Tissues are groups of similar cells that work together to perform a specific function. For example, muscle tissue is responsible for movement, while nervous tissue transmits signals throughout the body. Histology allows us to see the intricate details of these tissues under a microscope. Imagine being able to zoom in on a seemingly insignificant speck of skin or a tiny piece of an organ. Histology makes this possible, revealing a hidden world of cells, each with its own unique structure and purpose. By examining these cells, we can learn a great deal about the health of the body. Histology is an essential tool in many areas of medicine, including pathology, the study of disease. By examining tissues under a microscope, pathologists can identify abnormalities that may indicate a disease process. This information is crucial for making accurate diagnoses and developing effective treatment plans. Cancer, a disease characterized by uncontrolled cell growth, often leaves telltale signs in the body's tissues. These signs are invisible to the naked eye, but can be detected through the careful examination of histological samples. When cancer develops, cells begin to divide and grow uncontrollably, forming a mass of abnormal cells called a tumor. These cancerous cells often display distinct changes in their size, shape, and organization compared to healthy cells. By examining biopsy samples, which are small pieces of tissue removed from the body, histologists can identify these cancerous cells and determine the type and stage of cancer. This information is crucial for guiding treatment decisions and predicting the likely course of the disease. The process of preparing histological samples for examination is a meticulous one, involving a series of steps designed to preserve the tissue's structure and highlight important details. The journey begins with the collection of a tissue sample, which can be obtained through a biopsy or during surgery. The sample is then immediately placed in a fixative solution, which acts like a biological pause button, halting any further degradation of the tissue. The fixed tissue is then dehydrated, removing all water content, and embedded in a hard, wax-like substance, creating a solid block that can be sliced into extremely thin sections using a specialized instrument called a microtome. These thin sections are then mounted on glass slides and stained with dyes that highlight specific cellular components, making them easier to visualize under a microscope. Early detection of cancer is paramount for successful treatment and improved patient outcomes. Histology plays a critical role in this endeavor, often providing the first line of evidence that a tumor is present. When cancer is detected early, before it has had a chance to spread to other parts of the body, the chances of successful treatment are significantly higher. Histology allows us to catch cancer in its earliest stages when it is most treatable. By examining biopsies of suspicious lesions or screening samples, histologists can identify cancerous cells early on, allowing for prompt treatment and potentially life-saving interventions. The information gleaned from histological analysis empowers doctors to make informed decisions about the best course of treatment for each patient.